Yeah, good afternoon to all. Welcome to the second technical session of today's webinar, Emerging Trends in the Natural Products Research. Now I welcome Dr. Umarani, Department of Pharmaceutics, to chair the session. Welcome, Umarani.
Thank you, madam. It's my pleasure to introduce Dr. K.N. Chidambaramurthy. Sir is explaining about herbs as source for medicine's journey from bench to bedside. Sir currently working as Professor and Dean, Newburgh Anand Academy of Laboratory Medicine, Newburgh Anand Reference Laboratory, Bangalore. Sir has completed Bachelor in Pharmacy in Pharmaceutical Sciences, University of Mysore, and Master in Pharmacy, Pharmacognosy and Phytochemistry, Rajiv Gandhi University of Health Sciences, and Diploma in Research and Development, AIMAT India, and Doctor of Philosophy, Biotechnology from CSIR CFTRI. Sir has total 12 years of experience in India and in USA 5 years. Sir worked as Senior Research Fellow CSIR CFTRI India, and Sir worked as Postdoctoral Research Scientist and Associate Texas A&M University, and worked as Principal Scientist Ramaya Medical College, Bangalore. Sir has 4 patents to his credit and 117 research articles in International 78, National 15, and over 12 reviews. And to his credit, 2 books, chapters 15.
Hello.
And Sir has given more than 35 invited talks, national and international level.
No,
And
the
Sir
ಇನ್ನೊಂದು
has total
ಬೋರ್ಡ್
55 national and international presentations to his credit and conducted more than 24 research training programs. And Sir is the recipient of Dr. V.N. Patwardhan Prize
Okay.
2012, ICMR Government of India, and MNAMS from NAMS, New Delhi, and a Research Publication Award 2014, Government of Karnataka, and S.S. Mishra Memorial Award, NAMS, New Delhi, and Best Paper Award, 62nd Indian Pharmaceutical Congress. And Sir currently working as Professor and Dean, Newburgh Anand Academy. With this long profile, I request Sir to deliver his lecture. Welcome, Sir.
Thank you, madam. Hope I'm audible to all the participants.
Yes, sir.
Okay. Thank you. And before I start, I thank our senior, Dr. Sudha, who contacted me to give a talk. And we did post graduation from the same college. She's a 2 years senior to us. And the title of the symposia is an emerging trend on national product research. So morning we heard Dr. Mrutyunjaya talking about the application of alternative system of medicine, predominantly referring to natural medicine in the different part of the world, and what are the means to integrate that along with the mainstream, and how the entire globe is going towards accepting the alternate medicine and integrating the natural product research into a mainstream medicine. So, being topic being emerging trends in natural product research. So, what I thought was, let me give some insight based on my experience for last 20 years. I did my MPharm in 2001, and since then, constantly I've been working on natural products, whether in a hospital setup or in a R&D setup or in a research lab. So my main focus, although I work on different biological activity, my main focus is on exploring the benefit of herbs, diet, and other natural products, for mainly focusing into cancer. So this is the um, work I'm doing, and I would like to share some of my experience and views on why natural products can serve as a better alternate. So I'll share my screen. Yeah, I think my screen is visible for all the participants.
Yes, sir.
Yeah. So with this background, let us go ahead. So today's topic is herb as a source of medicine, journey from bench to bedside. Probably you've been listening to these titles from very long time, and a lot of people have made an attempt to convince that herbs as a source of medicine can work uh, for various ailment, and we always have a belief that herb or a natural product will have application only in case of chronic disease. They can't do the pharmacological action required for acute disease and other things. So before I start my presentation to all the young students who are aspirants of doing research and making their career from both pharmacy and research based thing, so I just want to take you through a journey where, and most of the views in the presentation is based on my opinion and as well as my research done so far. So I'm not either contemplating anything or telling that this is the good, and I'm sharing my views on certain topics where I have personally done a research. And this is something, let us start philosophical. After having a heavy lunch, and probably in Hyderabad, the temperature is shooting up. You all feel sleepy, especially if you have online session like this. So you switch on your screen. By seeing it, you go dose off. So before that, I just want to say that most valuable is least taken care of. So as a student, most of you, if you agree with that, I can give a small incident. Suppose if any of you uh, meet with, fall down or meet with a small accident, it is not that you are going to check what happened to your body. So that's for the newer generation. So first thing, even if you get up in the morning, it is not that you are looking for your health or anything. You care for your gadget. So even if you meet with an accident, first you look is what happened to my phone. Is it phone is all right, or my gadget is okay? So more than our heart or any of our internal organ, we give more importance to things which are least priority. So although now we are more dependent on cell phone, so we can't live even a minute without cell phone. So this has been our dependency. So the heart, which untiredly works for years together, we don't respect it unless we have a chest pain, or we have any issue with the heart. We don't go for checking the status. So I always say that philosophically, the most valuable things are least taken care of. So I'll come to the same anecdote at the end of my presentation to link what we are trying to learn today. And these are some of the information I want to share. So herbs are plant most promising source. The history has proven it, and we have a lot of evidence on that. And starting from the genesis of modern medicine, all the success has been seen in terms of herbs or molecules coming out of the herbs. So I'll just brief you on few of the things, and then difference in the journey. So when we have to work with uh, chemical or a drug molecule which is synthetic, what are the route we take and how different it is, sir. So there is a typo here. It is a phytochemical. Um, how different it is for herbs and chemicals, and what is the difference between a molecule and extract. Most often we come across this when we are working with natural product. They say the isolated molecule is more active. Then there is some a discussion which say the extract as a combination or cocktail of multiple compound will be more. So what we need to focus our work on. And since the word chemical sounds weird for some of the Westerners, they started telling these phytochemicals as bioactive molecules. Shortly, it is BAM. How they can help in chronic disease? What are the advantages? And yeah, we all know that nature, the best chemical engineer. So why, although we have advanced in chemistry, synthetic chemistry, and a lot of allied area, why nature is still remaining as one of the best chemical engineer? So we can't predict. And I want to give example of some work of mine on cancer and terpenoids, how the journey from lab to clinical study has happened, and also give some points and challenges when we are working with plant. Unlike chemical, So what are the additional challenges we need to address, and way forward, and a message to young researcher, like if you are interested to work on natural product, what are the aspects you need to, because the theme of the presentation or symposia is the emerging trends in natural product research. So I thought I'll justify by giving what are the missing link, focusing which you can see a better result for your research. And all of you agree that, so over 80% of the drugs used today, directly or indirectly, are generated from plant, directly from plant, or some are derived, and some are what we call as semi-synthetic derivatives, which are modified thereof, starting from aspirin and morphine, codeine, quinine, vincristine, vinblastine. Many of these, especially when it comes to cancer and respiratory disease, more than 80% of the drugs which even today they are used clinically are directly or indirectly derived from natural or herbal sources. So they have directly originated from plant or plant derived products, and sometimes because of the limited quantity they have been synthetically produced, and some are modified version. For example, morphine is further modified to get the better activity. So like this, so these clearly indicate that we can't say that herb or plant based medicines will not have value or they can't work. So most of the medicine even used in chronic and acute disease have, we all accept this. Most of the master student know about this. There is nothing new about it. So I link this to the current knowledge. As of today, approximately 70,000 species have been screened for utility and medicine, and this is the data which is pre-COVID era. So during the COVID, you'll be surprised, more than 1 lakh natural products and plants have been screened for its effect against the topmost search research they have done is antiviral activity. The second thing is to uh, immune boosting, especially on the surge of immune marker. So these are the two top research last 2 years. People are working in addressing the COVID. So more than 1 lakh or 100,000 plant or plant products have been screened. And finally, we got some good clinical leads in terms of benefit from ginger, green tea, and other things in addressing the secondary symptoms. Although we didn't get a molecule or a product which can 100% kill the virus, but we got a cocktail and some recipes which can uh, heal the COVID symptom faster, almost 3 to 4 days earlier. The recovery was seen in two of the clinical study where they use a ginger in one of the study, and other one is coronil, herb-based medicine developed by Patanjali. These were the two success stories from plant-based molecules. So about 8 out of 10 drugs used to treat the infection, cardiovascular disease, and cancer, as I told, and especially the immunosuppressives are from plant or some are from marine sources. And between, especially when we talk about anti-tumor and cancer-related drugs, so around 155 anti-tumor drugs have been developed into phase 3 clinical study, and some of them have gone for clinical application. All of them are almost derived, more than 50% say derived from natural products. So this slide clearly gives an importance of plant or plant sources as a potential source of all type of medication, starting from chronic and acute. But still, why they say that phytochemistry or pharmacognosy is losing its value? And there are, I mean, I often hear that we don't have admissions, we don't have people to take the pharmacognosy, or the trend of people opting for pharmacognosy is declining. That's not because of the science or nature. It is all because of the system what we are working on. It is more to do with the system of education rather than the uh, specialty itself. And see, again, these are some of the other drugs like For Alzheimer's, we have antiviral. I mean, many of the antivirals what we use clinically today are it may be podophylotoxin, or it may be some of the triterpenoids obtained from cannabis and its derivatives. Most of these are clinically proven for their efficacy, and their mechanism is established, and it's been approved by FDA and other international agency to be used as a therapeutic agent. So this speaks about there is a potential um, source from herb and herbaceuticals. Yeah. So apart from that, we also have traditional I mean, today morning, Dr. Mutunjaya clearly mentioned how traditional healing and healthcare has been integrated globally. We have a documented system. It is not that we say so many drugs are there. So traditional Chinese medicine has documented more than 47,000 herbs health benefits, and they have identified 25,000 pure compounds used, including their 3D structure, mechanism of action has been evaluated for various health benefits. And the herb information knowledge, the think herb database, which gives almost 500 drugs of natural origin, mainly from herbs or plant-based things. And they have given uh, the genes which are altered by this. They have pharmacological activity, entire PKPD has been explained, and they also give a signaling pathways in both human and rodents. So in addition to that, Indian plant is exclusively made for anti-cancer, because cancer is something which has seen endpoint in terms of synthetic because of the toxicity and adverse reaction and unexpected biological event. So the plant has been a successful source of positive outcome from last 3 to 4 decades. So therefore, there is a database which says this Indian plant anti-cancer database, which impacts provide all majority of Indian plant. More than 10,000 Indian plants have been uh, quoted with their specific activity and how they can address different types of cancer in human. So see, this gives us an important information that it is not that we don't have enough information, but we have it in certain form of textbook. So what we are trying to do is we are trying to see compare with the modern medicine. Like we have a molecule, we know the binding site, and this is the pharmacokinetics, this is the pharmacodynamics, and I have complete picture. So we are trying to match the herbal products or herbaceuticals or natural product into that. So actually we are comparing apple to orange, so we are not comparing apple to apple. The reason is the natural product, and most of the pharmacognosist sitting in this forum, and even Dr. Sudha will agree with me that plant produce these compounds are phytochemical, not for the human application. They are all produced for the protection of plants from the environment and the stress and I mean, biotic, abiotic, all type of stress. And nature has designed these molecules in such a way that these molecules, as long as they are in plant, they will be safe for the plant. They will not be toxic, but the moment they come out of the plant, even some of the compound, the structural modification happens, which becomes more toxic. And this is that's how we say most of these molecules are in certain forms of isomerism. Like we all know that many of these phytochemicals available inside the plant in leo form, when we extract it with the solvent and all, the um, optical activity changes and it becomes into dextroform. So that's why we all believe that nature has created in a special way where it can provide a maximum protection to the plant. But with our science and knowledge, we are trying to exploit that for our benefit. So when we are exploiting, we need to create and maintain the same environment till it reaches the human body for production of or producing the activity. But what happens during the course of transformation, we expose it to various changes. The solvent extraction, probably we make a mechanical engineer to the plant, and these are all lead to compromise in the activity, and finally we blame that this molecule not work. So it is other way. We need to understand the stereochemistry in the nature and then try to link. That is something which we need to strongly. So we also have international pharmacopoeia, Indian, British, and US pharmacopoeia, which clearly gives an information. And most of you, which you would like to, if you refer American Hub of Pharmacopoeia, America has spent close to 11 years to study only 4 or 5 drugs. One is digitalis, another one is turmeric, and two more drugs. They did a complete study on those things, and they have every piece of information related to that. So that is the depth of information we need to create to convince them. In addition to that, we also have a natural medicine comprehensive database, which is first edition is published in 2007 and revised in 2013, and this gives close to 400 natural products globally used. And yeah, coming to the journey of those who are in pharmacology and clinical pharmacy, you know this very well. So drug development has major 3 stages. So when we start with the drug discovery, we start with thousands of molecules, structurally similar compound and close compound. And then when we go to preclinical animal study, out of which only few hundred or few tens will come into the qualify that stage, and then we go for stage 3, and finally we get 1 or 2 compound for the regulatory approval. This is the normal process of drug development. The same thing when we working with a synthetic drug, we have to purify the compound, characterize using the advanced technique, and say that is 100% pure, because any impurity will cause a confusion whether the activity is due to major compound or impurity. And then we do activity screening, any pharmacological activity we would interested. Then probably we also study the structure activity relationship to establish that this is the structure that is required for receptor binding and producing the activity. Once that happen, then we select the most active form, and then we send it for activity screening. So this is the normal process which happen with the a synthetic molecule. But when you go to phytochemical, so it is little bit different. So first we need to gather an information either with a traditional knowledge, because traditional knowledge and traditional text are the resource for understanding the phytochemicals and its application for health. Because unless you read Ayurveda in depth, it is difficult to predict what will happen. Many of the plant we might have not seen it, because the change in the climate and ecosystem, some plants are vanished and we may not have. And if you refer to one of the traditional text, they also give the description of a plant and where it is available. And based on that, we need to identify the right source. And once we identify the right source, then we have to see where the plant is origin, like which part of the country or which part of the state the plant originally grown. And depending on the agroclimatic condition where the plants were grown, we have to go and collect and authenticate the sample. So we also know that many plants look alike, but chemically and pharmacologically the content is entirely different, because plants belong to Solanaceae family, will have certain set of characters. So there will be very close matching plants which are having exactly different phytochemical, and some may be toxic. So we need to have authentication based on the morphology, microscopy, and other characters. And then not only the plant source, but we have to also have authentic preparation, because when you refer to classical text, it is not the plant leaf or herb you root you powder it and directly consume. They have a process of preparation, either a maceration or a decoction or various other process. And sometimes they do a fermentation to concentrate and convert. So actually what today we call biotransformation was explained way back in 5000 years in India, where they have fermented many of these plant-based product to get an active principle. And then once we find out the authentic thing, we have to optimize the preparation, because unless you do the authentic processing authentic preparation steps properly, you don't get the optimum concentration of the active ingredient. Then after that, you have to screen and identify the marker compound. This is just to trace, have the traceability, and then we have to go for biological activity screening, then optimization of biomarker. For because unlike chemicals or synthetic molecule, we don't have a direct one receptor. We have to optimize set of biomarker which are responding for this particular decoction or herbal formulation. Then once it proves efficacy, then it will go for such 1,000 molecules can go for the study, and then they all pass through the regular. So this is the main difference between herbal drug and a chemical and a phytochemical when it go to studies, clinical studies. And the next slide explains you the approaches. So most of the industries will have this is the, again here also, if you see in the middle of the thing, Ayurvedic therapeutics, this is again based on the Ayurvedic usage and formulation. They have a central basis of Ayurvedic therapeutics. The script of Ayurveda is used here, and based on that we do either a reverse pharmacology, which most of the industries are following, or we use a route of ethnomedicine based on the ancient system. We understand their benefit, and by interpreting the literature, we go for activity screening, and finally we come up with either a molecule or a cocktail of thing. So this is the approach which we need to give for natural drug, unlike a synthetic molecule. Yeah. So with all these things, right now we are using the phytochemical or plant-based medicine only under certain condition. For not for all the disease, we opt for it where there is no treatment by modern medicine or chemically synthesized drugs, and treatment leading to toxicity and adverse reaction. For example, most of this cytotoxic drug used, chemotherapeutic drug used in cancer treatment, they have a narrow margin of safety leading to toxicity. So that time we think of herbal or plant thing. And whenever we need a long-term treatment, for example, disease like the chronic disease such as cancer or diabetic, where we need to consume regularly, then only we look for alternative means. That is when the herbs or natural products are gaining importance. And for an unknown disease, a disease with unknown origin like the COVID, when they started the initial phase, they approached lot of natural product to overcome this. And secondary effect from allopathy, there are some adverse reaction. What is it may be a hepatotoxicity or any chronic kidney disease or nephrotoxicity. Whenever we get these disease, so then we look for alternative treatment. It is not even now. It is not seen as a first choice for many of the disease. So unless we change our mindset to go and look plant or herbs as a first choice for disease, it will be difficult. So with this, I go to my research topics. What are the uh, research findings we have done, the rest of the world has done on cancer and herbs. And these are some of the molecules which are seen, like artemisinin, berberin, curcumin, EGCG, and cucurbit family ginseng compounds. And these are some of the compounds which are globally considered as anti-tumor drug, which are clinically proven. So we have used some of these in your model, and I worked on some of these compound. I'll just share few of them. Yeah. So when it comes to cancer, as I told you, there are very less choice in synthetic drugs. So eh, the use of herb has been always a choice, because we have shown the turmeric is an anti-inflammatory almost 5,000 years ago, and the antibacterial effect of tulsi and asimum is documented in our ancient literature. And also, the first natural molecule to complete the clinical trial was vincristine, and followed by vimblastin, which are in almost 1950, which is more 70 years back. We have shown the efficacy, and these are some of the drugs which even today, for example, taxine and icamantine, or even today the drug approved by FDA for use in cancer. These are all from plant source. So there is a lot of potential from natural or plant sources for addressing the cancer and related disease. So in that, I just give some of the example what we have worked with. And again, this is to show the efficacy of lot of these drugs like vincristine, camptothecin, which is obtained from Chinese tree, and we have podophyllatoxin, and you can see this. They all have a LD50 of 30 mg per kg, and as I told, compared to synthetic drug, the margin of safety is very big in this, and that makes them the clinician to use it comfortably to get the optimum activity. And curcumin is something which has been explored for last 2 decades for all type of cancer, and it did show a lot of benefit, especially in kidney disease, kidney cell carcinoma, and even in prostate cancer, where there are no much chemotherapy choices. So, and all of you know that these are the molecules which have that. So, Taxol, I mean, these are all the natural sources which have shown uh, their highest efficacy and been used clinically. Yeah, this is something which in 2003 article by Sur, who is a famous scientist from Korea, in his Nature review on cancer, he write about it is worth to study on these natural product for cancer inhibition. So, and he said the first successful neoadjuvant ingredient is turmeric. And in 1972, our own Indian CDRI, Central Drug Research Institute, has shown it as an anti-inflammatory. And in 1998, it was first time the clinical trial was done on a cancer patient. And total number of hit and the internet hit on curcumin and cancer is 74,000. So many people have searched, and so far number of clinical trial is hit is almost 32, and there have been more 300 plus. This is almost 5 years old data. So now it has been almost for close to 400 clinical trials of all phases of only cancer. I'm talking only about the cancer related thing, and majority of them were done in MD Anderson in Houston, where we did lot of study when I was doing my postdoc. Yeah, why these natural molecules are superior compared to thing. So as I told, the safety, relative safety, and their abundance, like it is there in the nature, we can always mimic and multiply it faster, and they always have a feedback mechanism. Unlike a chemical molecule, uh, these have a pattern, like to some extent they show the toxicity, and again after that a body has a mechanism of disposing this, because they are natural origin. This feedback mechanism make them wonderful molecules for cancer treatment, and non-toxic to plant especially and normal cells. So each time we want to test that the toxicity, 60 to 70% of death in cancer patient during the treatment is mainly due to toxicity to both affected and unaffected organ. So the toxicity is a general phenomena when it comes to chemotherapeutic drugs, especially synthetic. So there, these natural products always spare the normal or natural tissue from the toxic effect, and the complex in nature. So this complexity will have advantage in not producing more toxic to many of the organ. So because of the complex nature, its permeability is also limited. Sometimes the common toxic effect like crossing the blood-brain barrier and other things are taken care naturally with these complex molecules. So these are some of the thing, and this is the approach we all agree that we published in 2014. Like whenever we as I again told, this image is mainly based on the cancer, where we wrote like whenever we find a novel source based on the ancient system, we need to do a in vitro screening, and then using all the chemistry tool, we need to identify. It is not that we want to isolate a compound, it is to just ensure that what compound is present, so that when we make and give an extract, so we are able to trace the bioavailability and other stuff. Then we go for in vivo screening, and then the active extract will be directly, because majority of these, if they are used as a drug or a food compound, we can even bypass some of the toxicity studies based on the literature, and straight away it can be given for a trial, clinical trials, especially bioavailability and biosafety. So that is the advantage. This is the mode what they follow in use of cancer, I mean, natural drugs in cancer treatment. So this has been our uh, learning. Extracts are group of phytochemicals versus pure compound. The pure compound, the moment it is removed from the plant and separated from rest of the compound, it will have more of a synthetic molecular nature compared to a natural compound. So it is always better to work with the cocktail or a multi-constituent extract, which will have other ingredient which can synergize the activity. So that's been a learning for us. And molecule can target multiple pathways. So for example, if you take a diclofenac or paracetamol, they go and sit into substance P, and they have a specific binding site. Apart from that, they can't um, produce any other pharmacological act. They are confined to one. But the natural molecule, something molecule like a curcumin, which is a mm, low molecular weight molecule, can trigger multiple pathways. It will act on cell signaling pathway, apoptosis pathway, inflammatory pathway, and you name it, it will have one direct or indirect impact. So that is the advantage of natural product. And yeah, as I told, most of the time they can spare a normal cell or a tissue. The toxicity is very low. um, yeah, so it will also prevent the invasion and inactive circulating cells. So as we know that circulating tumor cells causing the metastasis is the major challenge in cancer. But most of these natural products, including turmeric, resveratrol, they have ability to combat or kill the circulating tumor cells. So this is a type of cell which will be circulating throughout the body, and they will spread the disease. So they have a very good inhibitory effect on circulatory tumor cells. So my uh, collaborator from Indian Institute of Science, Dr. Annapurni Rangarajan, works a lot on those things. We have a collaborative project. So those who are interested can read some of our article, which I can share with Dr. Sudha. So and then yeah, translation of activity from in vitro to in vivo, which much easier when we take a plant in case of cancer. And yeah, as most of these we have a complete profiling of the plant, and we also know a signature molecule to say that this is the authentic source. So some of these established plants are always easy to, these are some of the advantages using plant sources. And see, these are some of the herbs which have been clinically tried on regular basis. And the last one, the noni fruit, I probably a lot of you see in television nowadays. This is considered to be a wonderful fruit which has lot of effect on purification of blood, basically detox, and can prevent lot of cancer, at, especially organ specific cancer at very early stage, both colon and kidney cancer. It can prevent at very early stage. And we know the association of lycopene with prostate cancer. And green tea has shown effect on general inflammation and colon cancer. Both turmeric and garlic, along with ginger, are shown a very good impact on prevention and delaying the onset of colon cancer. So these are different clinical evidence we have in front of us. So toward this, and also there is a group which works on spices, which can prevent a lot of cancer early stage and uh, development stage of a tumor, mainly because of the volatile compound. And some of the other, some of the common things are cinnamon, mustard. These are some of the household uh, spices which have shown clinically efficient for inhibiting various cancer. Coming to the mechanism, as morning we were discussing the evidence why people are a little bit hesitant of accepting natural medicine is that they all want an established mechanism. They they won't be convinced if I say, okay, you take this juice or a plant extract for 1 week every day and your symptoms will come down. So everybody want to know what is the mechanism. So these are some of the common mechanism for prevention of cancer. Like lot of this plant will have antioxidant activity, then inhibition of cell proliferation, and sometimes they are detoxifying by activation of phase 1, phase 2 enzymes, and they also activate glutathione peroxidase, catalase, and these are the general thing in terms of prevention. And these are specific which can help in progression, including prevention of DNA binding, regulation of steroid hormone metabolism. So these are the mechanisms which have been proven for this. And in case of papillomavirus, the antiviral effect of some of this plant is known to be preventive. And see, coming to my research, so far I spoke about the information available. The actual research, what we have done is we have established a mechanism of cancer inhibition by some of the fruit and fruit derived compound, starting from pomegranate, grapes, and citrus is something which we have worked extensively, and also the compounds in watermelon. Some of the essential amino acids we have shown how it can help in cancer and also in cardiovascular disease. And in terms of herbs, we are currently working on some of these. The highlighted one, aloe vera. This is mainly to prevent the wound in cancer. Right now, we are seeing how the chronic wound can be prevented. The cancerous wound can be prevented, and we are working on Tinospora cordifolia. Simiruba glauca is one of my student who is working on the clinical efficacy in both radio protection as well as chemoprotective activity. And noni is something which we are working on both colon and colorectal cancer. So these are some of the clinical study, and apart from that, today I'll share some of the preclinical and cell line based studies which has enabled us to take it to the next level. And this is the compound which we have established a mechanism for first time. The triterpenoids, which we all know that abundant in nature. In these two triterpenoids, which are rich in Rutaceae family, and also the citrus, and it is also there in neem. The bitterness of neem is mainly because of these two compounds. And coumarins are a class of compound which are found in most of the diet as well as plants used as medicine. And monoterpenes, which are volatile and which give the fragrance, like d-limonene in lemon and citrus compound. And berberin, which is a yellow color alkaloid, has shown excellent activity on inhibiting colon cancer cell. And in terms of polyphenols, both curcumin as such, because we have a limitation of bioavailability in curcumin. So we have used the encapsulated curcumin using a natural nanoparticle. So that has helped a lot. And we have also shown to establish the relative activity, the structure activity of various flavonoids which are found in nature, to say that which flavonoid is good, especially for colon cancer. So yeah, this is the first piece of information. We have established how triterpenoid obequinone, which is present in both citrus family and neem, has shown. So basically, these are some of the Western blot result where we have shown how the proteins which are required to kill the cells or induce the programmed cell death has impacted the colon cancer cell using SW480 cells. And apart from one mechanism of apoptosis like intrinsic, they have also shown effect on extrinsic. There are two ways of receptor mediated and cell mediated. Both the mechanism these compounds have shown, and you can see the inhibition was seen in less than 50 micromolar. And in order to see whether the activity is uh, by the nature of a compound or it is just a toxic effect, we have also combined with the camptothecin, which is a chemotherapeutic drug, and we have seen that there is a synergy when we combine these two. And this you can see the publication for more detail. And then similarly, we have used the same compound obequinone for pancreatic cancer cell. Like pancreatic cancer is something which is a comparatively very deadly disease, which have a mean survival time of less than 6 months. And the pancreatic cells are difficult to address, cancer cells, because the access to the cell and some of the unique mechanism where it requires both anti-inflammatory and program cell death induction at a time. So we have seen that these obequinone can induce both apoptosis by multiple mechanism. At the same time, it will also act as anti-inflammatory. So this has given it a special quality, and we found that this can act in multiple pathways. And then I'm giving only few examples, like we have such several compounds we have elucidated, and from each category I'm picking one. And d-limonene, which is a flavor or a flavor compound present in monoterpene present in most of the citrus fruit. We have found that since it is a non-polar compound, we initially when we tried, the absorption was a challenge. So like when we tested the absorption, when we treated the cells with d-limonene, the entire plate was filled with the fragments, but the d-limonene was floating on the cell without entering into the water media. So we have to make it as a soluble emulsion. When we made it like a soluble emulsion, then we have ensured their bioavailability, and the same has shown uh, the induction of programmed cell death and also inhibition of VEGF, vascular endothelial growth factor, and which leading to um, our induction of apoptosis. So again, we have shown by 3D cell model that how the vascularization, the blood vessel formation is blocked by BEO is nothing but this one bioavailable volatile oil of the extract D-terpene. So we can clearly demonstrate that D-limonene can activate and inhibit the angiogenesis. Apart from that, we also seen MMP, matrix metalloprotein 9, which is very important for new blood vessel formation has been inhibited. So this shows that a compound, see for example, D-limonene is there in the peel of lime, and both obequinone and limonoids are present in seed and inside the fruit. So the whole fruit, if you take, it has different category of phytochemical which can act differently. Again, this you can see more detail in this article. Yeah. So those are about the citrus related compound. And curcumin, we all know that that's a gold standard for inhibiting NF kappa B. But the challenge with curcumin, there are two issues. One is whether the natural and synthetic curcumin will have the same impact. This is something for uh, most of you are wondering why we have to go for natural source when the synthetic source are available at much lower cost. This is turmeric we extracted, isolated from the regular rhizome of Curcuma longa, and we used synthetic curcumin, which is available readily from the one of the synthetic chemical company, so which is 100% curcumin alone. Unlike natural mixture, which will have curcumin dimethoxy and bis dimethoxy, and we did a study which when we use that molecule and did the activity, we found that the synthetic 100% turmeric or curcumin had a very low impact of NF kappa B inhibition. And again, nano curcumin absorption using caco2 cell, like to we know that there are two barriers. One is curcumin because of the structural nature and its uh, beta glutathione, or the one of the enzyme which is responsible for cleavage of this turmeric or curcumin, will reduce the absorption. In order to prevent that, we used a nano curcumin using a encapsulation of curcumin particles with whey protein, which is again a natural molecule. We never wanted to use a synthetic polymer to make a nanoparticle. So this is something which we did for the first time, and we could see like 4 to 5 times increase in the absorption. So these are the things, and we published the same thing in European Journal of Pharmacology about the mechanism. Yeah, again, this is something we worked out of our curiosity, where we wanted to see when we mix two well-established natural compound, whether we can see a synergy or not. So when both limonene and turmeric or curcumin were mixed, we could clearly see the synergy in their activity, and say that all the enzyme activity and the images clearly indicate when we combine more than one natural molecule, we can see a synergy in their cancer cell inhibition. And the same was published in Food and Function. And berberine, as I told, this is something which most of the time the teachers give us an example. This is an exception for the definition of alkaloid, which is both liquid as well as color. So this was basically used for dyeing the silk. So we found that there was a literature which says that the berberine can inhibit the tumor. So we wanted to show what is the mechanism. We used different types of cell line. I'm presenting only one SW480 cell line data here, and it showed that it can inhibit the cell cycle at various phases, especially synthase phase. DNA synthesis is strongly inhibited by this berberine. And apart from that, it will also work. See, this is impact of berberine on normal cell. So this is the graph, which the top one is the graph which shows inhibition of cancer cell, whereas the same in the normal cell of, even if you use a 200 parts per million, the inhibition was, or sorry, 200 micromolar, the inhibition was less than 20%. So that is the advantage with this natural molecule. And again, coming to the mechanism, it has shown uh, inhibition of proteins which are anti-apoptotic, basically inducing apoptosis, and also the VEGF was inhibited. And apart from that, the key signaling protein, survivin and other cell death proteins were also induced by berberine. And then and for the first time we also established a mechanism, like what are the different pathways a single molecule can act, like this is a receptor mediated apoptosis. We also have intrinsic inflammation, and apart from that, it can also act on a cell signal pathway. So all these mechanisms we could able to establish using a single molecule. And again, this was published in European Journal of Pharmacology. You can read this for more detail. Yeah. So I mean, those are with the pure compound. So when we are working in my lab, I always used to tell our students and friends, like, let us take one natural molecular group and say how SAR they do for a synthetic molecule. Let us establish. So then we selected flavonoids which are structurally similar and which are close by and which are abundant in nature, both in food and natural products like hesperidin, naringin, and naringenin. These are the flavones with slight change in their functional group, and one representative of flavone, that is epigenin, which is most potent tumor cell inhibitor, and four flavonols were also taken. So here we wanted to see how these closely related compound act on cancer cells. So these are the uh, proteins which were established, and out of the eight flavonoids we used, we have seen an excellent activity in quercetagetin and epigenin. These are the two which had shown highest induction of apoptosis, and the same both in Western blot and qPCR reaction. We could show that these two are very efficient. And yeah, the same thing. So we propose that if you are looking for a colon cancer inhibition, it is always better to go for natural sources which are rich in both quercetin and epigenin than any other flavonoid. Yeah, so these are some of the example which we were trying to establish, and like this we did lot of studies, and few of this we also took it to clinical study based on this information, as most of them are from the diet or food sources. Some without addressing the toxic issue, we could directly take them into a clinical study, and we have done two clinical study, one on a breast cancer, another one on a colon cancer, using all these terpenoids and uh, limonides, how they can help in preventing not only the tumor size, inhibiting the tumor size, but also help to overcome some of the adverse reaction from radiation. So what are the approach required? So basically, whenever we are working on natural product, all the screening should be done using pharmacodynamic basis, like to establish the mechanism. So probably using a high throughput screen technique will be one of the promising approach, because we have thousands of molecules to screen, and instead of screening for the activity, better we go for impact on each gene. We select set of gene panel and see how it can interact with each of them. And once we find out particular compound is very potent, then we need to look for enhance the content. For example, in case of flavonoid, we found epigenin and quercetin are very active. Then we need to look for breeding or biotechnologically tool within the plant, how to increase the content of these two compounds specifically, and use of extract for activity, and then only identifying the compound for qualitative purpose or for just marker and QA. And as far as possible, we have to use a minimum chemical or solvent. Even if you use them for extraction, make sure that their traces are not there when we use it for study, so that there is no interference from the other source. And as much as possible, we need to use a natural stabilizer instead of using preservatives which are chemically made. We need to use a natural stabilizer and a preservative. And the major focus should be on absorption and bioavailability. These are the two key parameter which can prevent any natural molecule passing to the next level. Either there is a delayed absorption or poor bioavailability. This is the bottleneck for many of the phytochemicals. We need to work on different model and improve these qualities. So, and then also with recent observation, the phytochemical interaction within the phytochemical, for example, phytate, the presence of phytate, some or some inorganic salt like calcium and iron, they have a chelating effect, and sometimes it is a inhibition through enzyme, how the phytate bind and make a complex with key metal ingredients. So we need to also look into phytochemical interaction. And equally, we have some of the compound which have a synergy. The classic example is how the bioavailability of iron is increased by ascorbic acid or vitamin C. So like that, we have lot of natural molecule which can synergistically act, and more focus should be on prevention, supportive care, and quality of life in terms of cancer. Instead of we try to screen molecule for tumor inhibition, suppose if we don't see whether it can also support the prevention or the means to improve the quality of life, either by reducing the side effect of radiation or chemotherapy. These are some of the areas which we need to focus on. Yeah, these are some of the uh, when we work closely with the cancer patient, these are some of the common adverse reactions they see: the stress, loss of appetite, anuria, and the pain in the body, unexplained pain in the whole body, and recovery, the post surgical recovery, detoxifying, sedative, and mood stability. These are the common ingredient which we need to give. So any natural molecule having these activity will be a good adjuvant therapy for cancer treatment. Yeah, and also anxiolytes, which are very important. And these are some of the barriers we see. As I told, these are source specific. Turmeric grown in the same place with different agriculture practice will have different content of curcumin in them. Again, the content in the primary source will matter, and the food or bioactive processing or manipulation of this compound is also important. And again, we need to also observe food and food drug interaction. Like we have drug interaction, we have some of the food they consume will also interact with the ayurvedic treatment. That's why most of the ayurvedic or herbal treatment are recommended along with the diet. So we need to take care of this. And we should always ask the question, can synthetic source slowly replace a natural one? Yeah, this is the last part of my presentation where I have, based on my experience, we have uh, listed some of the parameters which will have. Suppose if you are trying to consume a whole herb or a plant directly instead of using them as a medicine, what are the barriers? And people say that I eat that every day, but still I didn't get the benefit. So to address that issue, we wrote this chapter, challenges in optimal utilization of bioactive molecules clinically. So here we are bringing out all the parameter that will decide the bioavailability of or activity of bioactive molecule. These are plant related. Again, plant variety, agriculture practice, the soil type. See, this will all influence the accumulation of active principle or bioactive compound. And then the handling and processing will either reduce the content or they modify them into a form which is not therapeutically active. And then the last thing is the consumer or human factor, the health, the gut disease, any disease in gut or mutation in the metabolic enzyme and physiological condition and psychology of the patient can directly impact the benefit they are getting from this natural product. These are some of the general aspect, but more specifically, these are the factors which we all know. It holds good for both the natural and synthetic molecule, exogenous and endogenous factor, and route of administration will all decide the fate of our natural molecules and their biological effect. So you can read this more detail. Those who are interested can refer to this chapter, and you get more information. We have discussed on with example how each uh, parameter will help, and some of the examples known for day to day life. With this, I just want to say, before I conclude, I want to give some of the areas where, if you're planning for research on natural product, what are the potential area. So need for research at the institution level, and we should always establish that research what we do at the college or institution should also complement the industrial or scaling up need. So towards that, identify the plants or herbs used by local medicinal practitioners. See, we all say that okay, we go for internet and search for a lot of literature to say which is more potent. Instead of that, we should also focus on low, because as we know, some of the local traditional medicine practitioners will have a tremendous information on some of the plants. So identifying the local medicines available and talking to practitioners to identify the potential herb will be a primary and most promising thing. It can sometimes do wonders. Like the example is Phalanthus nererae, which is, it is even today a drug of choice or a treatment of choice for jaundice. So you don't have any other standard treatment for jaundice. So that comes through a local practitioner knowledge. So then document the local medicinal plants and herbs available throughout your area. These are, I feel, at the undergraduate level, these should be given as a topic to tap the local resources and undertake a translational studies to establish the mechanism. Because you can't say that I'll work, I belongs to pharmacognosy, I work only on this and extract. No, you collaborate with your friends in pharmacology, chemistry, and pharmaceutics, and try to address all the aspects for each molecule to establish the mechanism, and focus on ethnopharmacology studies, so traditional along with the modern pharmacology, and continuous research to take the potent molecule or herb or formulation to next level. What happens if some potent molecule or herb is available locally, it will never reach international researcher to take it to next level. So try to collaborate and work with a bigger lab within the country, because we have also have an IPR issue. So considering that, you collaborate with, because Hyderabad is such a place where you have several national labs. India's best pharmaceutical companies are located in your city. So make use of that, and try to take the help and work on translational challenges in collaboration. Like for example, bioavailability and some of the absorption related issues. Try to address this in collaboration with the industry. Then application of novel formulation and development technology to enhance the efficacy. Always talk to your formulation expert colleagues who can help you to make it more accessible by the cell, and increase develop a formulation which is more suitable for particular application. And yeah, create an evidence on safety of clinical studies. So before they go to clinical study, they always looking for safety either through the research or with the literature. Establish safety for each herbal drug or traditional drug that is been used. Focus your research with collaboration and integration, which definitely, because never think, because my most of my research, although I did MPharm in phytochemistry, I realized that my collaboration was maximum with biotechnology, biochemistry, phytochemistry, agriculture school, and medical college. So these are the things which made many of my research project and papers possible. So please collaborate and integrate for better outcome. So with these few words, I summarize my presentation, saying that plants and herbs offer a great potential as a source of bioactive molecules, more in cancer related research. Understand the application and limitation for use of plant source directly is critical. So we need to know whether we can use the plant as such, or in combination, or make a formulation out of this. This is very critical. That comes only through referring to a traditional practice or an ancient literature. Studies on herbs or plant with a modern pharmacological approach is more meaningful than going back to the olden approach. Then application of latest technologies towards enhancing the efficacy and bioavailability is critical. And finally, I want to say that tap the local traditional knowledge and wisdom. So it may be answer for your tomorrow's healthcare challenge. So with this thing, so again, coming back to most valuable is least taken care of. So we know that this is something like your heart, which always works for you. Nature has given a lot of things for you. Please, instead of depending more on a synthetic and exogenous source, try to depend on the nature, where we told, like, again, we need to respect nature and try to understand it for better application. With this, I would thank all my teachers and research mentors, and those research collaborators across the world, like the US, Korea, Japan. We have from different place all my collaborators, and my student and the research team who is constantly helping me to come up with new knowledge. And I also thank the management of Newberg Anand Academy for giving me an opportunity. And I especially thank Dr. Sudha, my senior, who invited me for this presentation, and the principal and faculty of the institution, along with all the student of RBVRR Women's College of Pharmacy. I would like to thank for attending my presentation. Hope my presentation could make some sense, and I am happy to have few questions to learn together.
Thank you, sir.
So if there are one or two questions, I'm more than happy. If they have written something in the chat box, you can just tell.
I hope everyone is clear with the presentation, I think, sir, actually.
Okay,
There
ma'am.
are no questions.
Okay.
Uh,
And if anybody
so,
want to contact, yeah, you're more than welcome.
sure, sir. Sure, sir. Sir, from my side,
Yeah.
what are the difficulties we find during isolation and extraction of medicinal compounds to maintain their potency?
No, I
Uh,
mean, my entire argument is don't try to start your work with isolation and extraction. First, try to
okay,
follow the ancient system of formulation, decoction, whatever it is. We should do isolation and characterization only after ensure the reverse pharmacology. What is practice? Try to first examine a natural product in the ancient way they are using, and then try to see whether it is for a quality check you make a purification and separation. Otherwise, if you can establish the activity in the extract or the formulation where it is classically mentioned, I think we can scale up that and achieve much more. So what we do is, instead of that, we try to separate the compound, purify the compound, and we get a pure molecule and say that finally it is not active. And most of the plant, based on my research, as a single compound they are not as active as the extract, the whole compound. So it is a combination of multiple compound in extract which make it more potent. That's why when you see olden
yes,
literature, you see a kashaya decoction, it is not a single compound. So you do the activity, establish the pharmacology, and then go for characterization. I think that will be a better approach than to trace a extract. You can have one or two marker compound. That is
okay,
more
sir.
meaningful.
Already
Yeah.
pharmacological studies have been reported. So we want to go for the next level of stage, suppose. So what kind of precautions we must take to maintain its activity and potency?
No, no, that's what. Like what now? I mean, I've seen more than 100 dissertation on both pharmacology as well as phytochemistry. What they do is they take a plant, they extract using a oxalate or something with a polar and non-polar they try to segregate them depending on the polarity, and then try to study the activity, which will never work. But if you go to the ancient literature, they always use either a alcohol or water as a source, and they try to extract. So what I request you to do is, whenever they use a plant, say for example plant X, you see the literature, what is the form it is used and what is the preparation they have made. Sometimes if it is fat soluble, they would use some ghee or other things for extracting the content. Example, phytosterols comes only in lipid base. So they, and if you take the same thing in n-hexane or chloroform, the chemical structure will be different and you don't get the activity. So the ghee is also non-polar and NXN is also non-polar. In lab, we don't use ghee because it is authentic. Literature say that take it in a ghee, but we don't. It's a fat soluble. We go for chloroform or NXN. So you can see the difference. But when we do the activity with the extract with NXN or chloroform, there will not be any activity because it also need a fat base to integrate and make the absorption happen. So if you extract in hexane and reconstitute in water again, the absorption will become. So there are small things. When you
Yes,
dissect the science behind the ancient knowledge, definitely you can make more sense.
obviously, sir. I agree, sir. So we
Yeah.
have to look back into our traditional methods like
No, basically
application
you
process.
follow, add traditional method, the science to traditional method and extrapolate, but not that make the traditional knowledge to your convenience and trying to
Yes,
extrapolate the knowledge. So most of the time, I think that is the critical point where we miss
yes, sir.
the opportunity.
Yes, yes, sir. There are no questions, sir, till now.
Yeah, thank you, ma'am. Thank you again.
Thank you so much, sir, for sharing the information. And we are actually very much fortunate with your great profile. We have very much inspired also, like the patents and the research profile we have seen. And the lecture was very interesting. And basically, I'm a Sutics person, but very much interested towards natural medicine.
Yeah. No. I always tell the student, see, our education is like a passport. It is not a visa.
Yes,
So whether your Sutics, your cognosy or
yes,
pharmacology, it is only passport. It is up to
yes,
you whether you want to reach Nepal
yes,
or
yes.
Mars. So
We
use
have
as
to.
a background and try
Mhm.
to do what is required for the
Yes.
mankind. Thank you, ma'am.
Thank you so much, sir. Thank you so much
Thank
for
you.
sharing the information,
Thank you.
providing the data regarding your research work. Thank you so
Sure.
much, sir.
Thank you.